The human digestive tract is a vast, interconnected microbial ecosystem. This environment functions as a nutrient circulation system, where shifting bacterial populations dictate the state of our systemic health. Our overall metabolic and immune stability depends on the balance of these microscopic communities. When these populations are in equilibrium, the body remains resilient. Diets high in saturated fats disrupt this equilibrium. These fats act as a form of toxic runoff, creating stagnant conditions that stall the natural nutrient flow of the intestinal environment. Under these conditions, a bacterial group called firmicutes multiplies rapidly. This overpopulation creates dense, static clusters that extract excess energy from food, contributing to obesity and metabolic dysfunction. Correcting this congestion requires more than just introducing new bacteria. It requires a targeted intervention that can alter the physical and chemical conditions of the gut itself. Polyunsaturated fatty acids, specifically the omega-3s, EPA and DHA, provide this intervention. In the digestive tract, they function as specialized, nutrient-rich currents moving through the ecosystem. EPA and DHA do not simply replace existing species. Instead, they change the ecological environment, creating conditions that naturally support beneficial bacteria while making life difficult for inflammatory ones. These omega-3s act as ecological regulators. By shifting the internal environment, they pull the gut out of stagnation and restore a thriving biological balance. As omega-3 nutrient streams enter a congested ecosystem, they begin a targeted restoration process. These currents actively suppress harmful pro-inflammatory species like disulfovibrio. By improving water quality and reducing toxicity, the omega-3s make the gut inhospitable to these harmful microbes. In this comparative chart, the omega-3 currents reduce the oversized firmicutes populations that drive metabolic imbalance. It is the primary barrier separating the dense microbial populations of the gut from the sterile environment of the bloodstream. When this protective wall weakens, bacterial toxins, known as lipopolysaccharides, or LPS, can leak directly into the blood. This leakage triggers chronic, low-grade inflammation throughout the entire system. Omega-3s neutralize this threat by boosting the production of intestinal alkaline phosphatase. This enzyme acts as a local filtration system, detoxifying LPS molecules before they can cross the barrier. Omega-3s also reduce levels of zonulin, a protein that usually causes the connections between intestinal cells to loosen. Lowering zonulin keeps these cellular walls tightly sealed, preventing pollutants from escaping into the blood. By physically securing this perimeter, omega-3s prevent localized digestive stress from spreading and becoming systemic inflammatory disease. In this fortified, highly organized ecosystem, the gut can finally perform its most advanced and essential metabolic functions. When omega-3s are present, the gut can process soluble dietary fibers more efficiently. Fatty acids optimize the environment, allowing bacteria to ferment these fibers at a much higher rate. This fermentation process produces short-chain fatty acids, or SCFAs. These compounds, specifically butyrate, propionate, and acetate, serve as essential stabilizers. Butyrate acts as the primary fuel source for colon cells, providing energy to repair damaged tissue and prevent intestinal wall erosion. While butyrate stays in the gut, acetate and propionate enter the bloodstream, reaching distant organs like the liver, regulating systemic inflammation and energy metabolism. These signals eventually reach the brain via the microbiota gut-brain axis. Here, they help calm the body's primary stress response system, the HPA axis. Under chronic stress, problematic bacteria can interfere with the way the body processes tryptophan. Omega-3s suppress these species, ensuring tryptophan produces mood-regulating serotonin rather than inflammatory stress signals. By stabilizing the microscopic conditions of the gut, omega-3s maintain the biological pathways necessary for long-term metabolic health and cognitive resilience.